Thousands of chemical reactions takes place in the cells of living things. Most of these chemical reactions cannot take place at the temperature and the pressure at which those living things are living. So most of these reactions are unfavorable at those conditions. So how do these reactions they take place within the cells of these organisms? That is by means of the enzymes. Enzymes are biological catalysts. They carry out chemical reactions by decreasing the energy of activation. And in this way, they increase the rate of reaction. So in the absence of enzymes, almost all of the chemical reactions taking place in the bodies of living things will become unfavorable and hence cannot be done. Enzymes are protein in nature. They are made up of amino acids. So the substrate molecules or the reactants, they bind to a certain region of that enzyme, which is of course made up of amino acids, and eventually is converted into the product. That part of the enzyme where a substrate molecule or a reactant molecule binds is called as the active site of that enzyme. Some enzymes can have one active site, others can have more than one active sites. Many enzymes carry out chemical reactions on their own, but some enzymes need other substances which are called as cofactors. If the cofactors are small organic molecules which are dissociable, they're called as coenzymes. For example, NAD, which is nicotinamide adenine dinucleotide, is a cofactor for dehydrogenase enzymes. If the cofactor is tightly bound by the enzyme, then it is called as a prosthetic group. Although hemoglobin is not an enzyme, but the heme part of hemoglobin is very tightly bound to the globin protein. Hence, it is regarded as a prosthetic group. Similarly, for several enzymes, biotin is covalently bound to the lysine residues of the enzyme, and hence it is tightly bound to the enzyme. Some enzymes they need metallines as a cofactor to carry out chemical reactions. For example, the nucleic acid dependent enzymes they require magnesium ions to carry out chemical reactions. For example, DNA polymerase, nucleases, and other nucleic acid dependent enzymes, they need magnesium ions as cofactors. In the absence of these metalline cofactors, the enzyme will not be able to carry out its function. This is a table which shows different types of coenzymes and for the chemical reactions in which they participate. You are required to remember these coenzymes and the reactions which they carry out. For example, biotin is required for carboxylation reactions. Vit- Cobalamin, which is derived from vitamin B12, is important for alkylation. Coenzyme A is needed for the transfer of acyl groups. Flavin coenzymes, including FAD and FMN, which are flavin adenine dinucleotide and flavin mononucleotide, they are needed for redox reactions. Lipoic acid is also needed for acyl transfer. Nicotinamide coenzymes, which include NAD and NADP, which are nicotinamide adenine dinucleotide and nicotinamide adenine dinucleotide phosphate, NAD and NADP. They are also needed by the dehydrogenase enzymes for redox reactions. Pyridoxal phosphate is needed for the transfer of amino groups. Transaminases are the enzymes which carry out the transfer of amino groups. These enzymes they need pyridoxal phosphate as a cofactor. Tetrahydrofolate is a cofactor which is needed for the transfer of 1 carbon units. This include methyl groups, methylene, formyl groups. So 1 carbon groups are transferred by tetrahydrofolate. And thymine pyrophosphate is important for the transfer of aldehyde groups. These coenzymes are derived from the vitamins. Vitamins are those substances which are needed by our body mostly through the diet, and they are required in generally in a small amount. They themselves are not important for energy production, but they are needed by enzymes as a cofactor or a coenzyme. So the vitamins, they're precursors to coenzymes, and a deficiency of a vitamin results in some disease. So therefore, they're although required in a small amount, but they are needed by the body. This table shows the classes of enzymes. Enzymes are classified based upon the type of chemical reaction which they catalyze. Based upon this, there are six classes of enzymes, which are oxidoreductases. These are the enzymes which carry out redox reaction. Transferases, as the name say, these are important for the transfer of a group. For example, we have just talked about transaminases. These are enzymes which transfer amino groups. The third class is hydrolase. Hydrolases are those enzymes which carry out hydrolysis. The next one are lyases. Generally, these are enzymes which result in breaking a chemical bond. Isomerases are those enzymes which carry out the process of isomerization. And lastly, ligases. Ligases are those enzymes which carry out formation of a bond. For example, you would have studied about DNA ligase, the enzyme which joins the Okazaki fragments during synthesis of DNA. So these are the six classes of enzymes, and these names, they are in a sequential order. So class 1 is oxidoreductase, 2 is transferase, 3 is hydrolase, 4 is lyase, 5 is isomerase, and 6 is ligase. You are required to remember these six classes of enzymes, and you are required to remember them in the correct order. We will now have a look at the mechanism that how enzymes carry out chemical reactions. So before talking about the participation of enzymes to carry out chemical reactions, let us first have some basic information in this regard. This figure shows the reaction coordinates on the x-axis and the energy on y-axis. This is the substrate represented by S, which is the reactant, and this is the product which is produced after the chemical reaction. So the transition from S to P is a chemical change. In order to carry out this chemical change, certain amount of energy is needed, which is represented by this peak on the graph. This energy is called as the energy of activation. Energy of activation is the energy which is required to achieve the transition state. Transition state is an intermediate form during which the chemical change is being brought about. So when the transition state is achieved, the species is called as the activated complex. The activated complex or the transition state can transition both back to the substrate or forward to the product depending upon the conditions and the suitability. What enzymes do is that for reversible reactions, the forward and reverse reactions are in equilibrium. The enzymes do not affect the equilibrium constant. The enzymes only affect the rate of chemical reaction at which the reaction is being carried out. But once equilibrium is achieved, the enzyme do not affect the equilibrium constant for that reaction. So enzymes only affect the rate of reaction, but they do not affect the reaction equilibrium. How enzymes do this job is by decreasing the energy of activation, which is needed to carry out a chemical reaction. Enzymes interact with the substrate, and through different types of interactions, they participate in providing the energy of activation. So therefore, the total amount of energy needed externally is very low. So the energy which is released by these interactions of the enzyme with the substrate, that energy is used to achieve the transition state and hence carry out the chemical reaction. So the enzymes, how they increase the rate of reaction is by decreasing the energy of activation. This is a diagrammatic representation which shows the change in energy. On the x-axis is the reaction coordinate, and on the y-axis is the free energy. S represents the substrate, which is the reactant, and P is the product of this chemical reaction. For the uncatalyzed reaction, this black line shows the transition state. So you can see that from this ground state, a lot of energy is needed for the uncatalyzed reaction to achieve the transition state. How this energy of activation is provided, in many cases by increasing the pressure and temperature, the energy of activation is provided. This blue line shows the energy difference for the enzyme catalyzed reaction. So this is the transition state which is achieved through the enzyme catalyzed reaction. So you see that the difference from the ground state to achieve the transition state is very small in terms of energy. So therefore, this reaction can take place very easily. However, for the uncatalyzed reaction from the ground state to the transition state, there is a very large difference of energy. So this reaction cannot take place uncatalyzed. Now, how enzymes they decrease this energy of activation? Enzymes form multiple non-covalent interactions with their substrates. We know that when interactions are formed and bonds are broken, energy is released. So enzymes form multiple non-covalent interactions with their substrates. These interactions provide sufficient amount of energy which is used on site by the enzyme to carry out the chemical change and achieve the transition state. So enzyme they decrease the energy of activation by providing energy through these multiple non-covalent interactions. The non-covalent interactions, they are dynamic. They can be formed and they can be removed. These non-covalent interactions include hydrogen bond, ionic bond, hydrophobic interaction, van der Waals interaction. So these are some of the underlying principles that how enzymes they catalyze chemical reaction. The first important thing is formation of weak interactions. Enzymes interact with their substrate and they form multiple non-covalent weak interactions with their substrates. These include hydrogen bond formation, ionic interactions, dipole and van der Waals interactions, hydrophobic interaction. So collectively, although these are weak interactions, they are non-covalent interactions, but the enzyme establish multiple weak interactions. So a number of these weak interactions are produced, and the net result of these non-covalent interactions, since they are multiple, so the amount of energy they contribute is very significant. One non-covalent interaction has a less amount of energy, but when the enzyme forms multiple such weak interactions with the substrate, that contributes significantly to providing energy to achieve the transition state. Another important thing is that the enzyme interacts with a substrate and it specifically stabilizes the substrate in the transition state. The transition state is a state where the chemical change is being brought about. This is a figure from Lehninger principle of biochemistry. This figure shows the catalysis by a hypothetical enzyme. So this is the case where no enzyme is present. So the substrate in this arbitrary example is a metal stick, and the chemical reaction is to break the stick into two parts. So the transition state will be bending of the stick as shown here. In the absence of any aid, which is in the absence of enzyme, a huge amount of energy will be needed to bend this stick and hence to carry out this reaction. However, the enzyme decreases the energy of activation, but the enzyme is not complementary to the substrate, but the enzyme is complementary to the transition state. So in this illustration, you can see that the enzyme is complementary to the substrate, which is the metal stick shown here. Now, this is the transition state, which is the bent form of the stick. So in this form, you can see that it is very difficult to bend the stick because the pocket where the substrate is binding, that cannot accommodate the transition state. So again, the energy required will be very high. In the third case, you can see that the shape of the enzyme, well, the magnet shows the enzyme here. So in this third case, you can see that the shape of the enzyme is such that it can bind to the substrate in this form, but it also allows the substrate to undergo the transition state. So now the active site or the binding pocket is such that it can accommodate this structural change in the substrate. And hence, when the structural change is brought about, the substrate will be converted into product, because this structural change brought about in the substrate results in achieving the activated complex or the transition state. Hence, this results in stabilizing the transition state. And when transition state is stabilized, the reaction can proceed in the forward direction easily, with requiring less amount of energy. So another important thing is that the shape of the enzymes is such that they bind to the substrate preferably, and then they stabilize the transition state so that the reaction can take place in the forward direction and the substrate could be converted into the product. Enzymes are also important for maintaining a proper orientation of the substrates in order to carry out a chemical reaction. This is an example which shows this phenomenon. You can see that this is a chemical reaction, a condensation reaction taking place between these carboxyl groups to form an anhydride bond here. So if these two molecules are two separate molecules and they undergo chemical reaction, now these molecules are present in the solvent and they are free to move around in any possible given orientation. So therefore, there are less chances they're going to approach each other in the given proper orientation to form this bond. So therefore, the rate of reaction is relatively low. Let us look at the other example where these groups participating in the chemical reaction, they are sort of fixed by means of this carbon backbone here. So when these groups they are immobilized, and now both of these groups they lie on the same molecule. Hence, these groups they are relatively immobilized. So therefore, the rate of reaction in this case is more, because these groups they now lie close to each other in the appropriate orientation. However, since these are single covalent bonds, so rotation across these bonds is also possible. So if this background skeleton of the molecule is replaced by a rigid structure so that no rotation can take place here, so now these groups they are in a fixed position, a fixed orientation, and their orientation is not going to change. So when this bond is going to form between these reacting substrate functional groups, the rate of reaction is now much greater. So this, although this is a non-enzymatic reaction, but this example signifies the importance of proper orientation. Enzymes they bind to the substrate and they immobilize the substrate into a proper orientation where the substrate groups they can undergo a chemical reaction. That's another very important thing with the enzymes too. Enzymes they reduce the entropy. The molecules are present in aqueous environment in a living thing, in a cell, and these molecules they are able to diffuse in everywhere in the environment of the cell, and they can be in any possible given orientation and conformation. So therefore the entropy, or in other words the disorderness, is very high. In order for a chemical reaction to take place, the functional groups must be aligned in a proper orientation so that the reaction can occur. So this is done by binding to the enzyme, and the enzyme decreases the entropy, and hence it orders, it binds, and orients the substrate molecules in proper orientation where it can result in a fruitful reaction. So therefore, the rate of reaction will increase. Many of the substrates, they're bound by solvent molecules, and the solvent molecules, they can affect the chemical reaction. So in many cases, the solvent molecules are to be removed from the substrate molecules so that the chemical reaction can take place. Another important thing where the enzymes do is to carry out desolvation of the substrate molecules, and hence they remove the water. Well, in some of the reaction, water molecules, they do participate, but in many reactions, water molecules do not participate. So therefore, the enzymes, they act as a filter to remove the solvent molecules from the substrate so that the substrate can now take place in a given chemical reaction. The active site of the enzyme is not a rigid structure. The enzyme itself is flexible. The structure of the protein, the backbone of the protein, is flexible. So when the substrate binds to the enzyme and the transition state is achieved, which is then followed by conversion into the product, the enzyme also undergoes a little structural conformation. This change in the enzyme is to accommodate the transition state and hence to convert that transition state into the product. So the active site of the enzyme is a dynamic structure. So because of all these different features and because of these different chemical bases, the enzymes, they favor a given chemical reaction in the forward direction, that is from the substrate to the product. And by increasing the probability of conversion of the substrate into the product, the enzyme increases the rate of that chemical reaction. The enzyme also contributes through the weak interactions to providing the energy of activation for a given chemical reaction. So through all of these different mechanisms, the enzymes, they contribute to increasing the rate of reaction for a given chemical reaction, and they decrease the energy of activation to achieve the transition state.